Hello learners, today we will discuss dark reaction. We know in the photosynthesis there are two stages. First one is the light reaction and second one is the dark reaction. So we are going to discuss dark reaction today, and this is also known as the Calvin cycle, then after the name of the scientist, and the C3 cycle. We will see why it is termed as C3 cycle. So the first we need to understand that in the photosynthesis, primarily there are two phases, and that is light reaction, light reaction and dark reaction. In the light reaction, photolysis of water is done, and here a molecular oxygen is evolved, and along with molecular oxygen, ATP and NADPH which is the reducing power, is generated. So in the light reaction, oxygen is evolved after the photolysis of water, and ATP and NADPH are made. Okay, at ATP and NADPH are used in the dark reaction. So in the dark reaction, carbon dioxide fixation is happened. Carbon dioxide fixation. So in the atmosphere there is carbon dioxide, and that carbon dioxide enters in the plant leaf through stomata, and it diffuses and reach to the chloroplast. And inside the chloroplast stroma, see, this is the chloroplast. So here grana thylakoid structures. Okay, so in the stroma, this is chloroplast stroma. In the chloroplast stroma, there are various enzymes which catalyzes the dark reaction, in which carbon dioxide is incorporated in the carbohydrate like glucose, fructose, sucrose, starch. Primarily in the dark reaction, atmospheric carbon dioxide is is incorporated in the organic molecule. Okay, so let's see the scheme of the dark reaction or the Calvin cycle. So Calvin cycle operates in the stroma, and the main enzyme we can say is the Rubisco enzyme. We are familiar with this enzyme. This Rubisco enzyme, which stands for the ribulose 1,5-bisphosphate carboxylase oxygenase. This enzyme performs both activity, carboxylation and oxygenation. In the C2 cycle or photophosphorylation, it performs the oxygenase activity, but here in Calvin cycle, where carbon dioxide is being fixed, this Rubisco enzyme performs the carboxylation activity. So this is the substrate molecule. This is the substrate molecule RUBP, ribulose 1,5-bisphosphate. This is 5 carbon compound, C1, 1, 2, 3, 4 and 5. So at the carbon first and fifth there is phosphate group, so it is termed as ribulose 1,5-bisphosphate. So this is the substrate molecule, and carbon dioxide is added in this molecule, which is catalyzed by this Rubisco enzyme. And after the incorporation of this carbon dioxide, 2 molecules of 3-phosphoglycerate is made. RUBP, this ribulose 1,5-bisphosphate, in the short RUBP. RUBP has 5 carbon, and after the incorporation of carbon dioxide, it becomes 6 carbon intermediate, 6 carbon compound, which is unstable and subsequently break down into the two molecules of 3 carbon compound, which is 3-phosphoglycerate. Okay, and after that, see, in the Calvin cycle, there are various reactions which we can categorize in 3 major groups. First one is the carboxylation phase, that is carbon dioxide incorporation, as we have seen here, catalyzed by the Rubisco enzyme. In the second phase, reduction is carried out. Reduction of the 3-phosphoglycerate into glyceraldehyde 3-phosphate, and in the reduction phase, ATP and NADPH is used. Here 2 molecules of 3-phosphoglycerate which is reduced to glyceraldehyde 3-phosphate, and 2 ATP is used, 2 ATP and 2 NADPH, 2 ATP, 1 ATP for each molecule of 3-phosphoglycerate. So this ATP catalyzes this reaction, and there is an enzyme phosphoglycerate kinase, and one more phosphate group is added, and there is 3-phosphoglycerate, and after adding it becomes 1,3-bisphosphoglycerate, that is at the carbon number 1, one more phosphate from the ATP is added, and after that NADPH reduces this 1,3-bisphosphoglycerate and converts it into glyceraldehyde 3-phosphate. Okay, and this glyceraldehyde 3-phosphate is converted into the dihydroxyacetone phosphate, and after this there is regeneration phase, the third phase of the Calvin cycle, where glyceraldehyde 3-phosphate and dihydroxyacetone phosphate unites to make various interconvertible compounds or interconvertible products, which are fructose, erythrose, xylulose, and glucose also. So in the regeneration phase, these intermediary compounds are made by the help of trans-aldolases and trans-ketolases enzyme, and the main thing is ribulose 5-phosphate has to be regenerated again to complete this cycle or to start this cycle again. So ribulose 5-phosphate is generated, and again 1 ATP is used to convert it in ribulose 1,5-bisphosphate, this one which is the substrate for the Calvin cycle, and this cycle continues time and again. So in the Calvin cycle, if we summarize, there are 3 phases: carboxylation phase, and where carbon dioxide is incorporated; reduction phase where ATP and NADPH is used to reduce the 3-phosphoglycerate, and the third stage is the regeneration phase, where ribulose 1,5-bisphosphate, that is RUBP, which is generated again to restart this cycle, Okay, so if we see the balance sheet of the Calvin cycle, see, Calvin cycle generates the glucose or other sugars. Okay, if we take example of glucose, so glucose has 6 carbon, C6H12O6. So to get one molecule of glucose, 6 carbon dioxide molecule has to be fixed via this Calvin cycle, okay, or C3 cycle. So 6 carbon dioxide and 18 ATP. See how see, if for the 1 carbon dioxide, if in this cycle 1 carbon dioxide is incorporated, so we can see for 1 carbon dioxide two ATP molecules here and one ATP molecules here. Three ATP molecules for one carbon dioxide. Three ATP molecules and two NADPH. Here you can say NADPH. Two ATP and two NADPH molecules are expensed. So for 6 carbon dioxide, there would be 18 ATP and 12 NADPH molecules used, and 12 molecules of water. So the C6H12O6 glucose is made, and side by side when ATP is used, so ADP 18 ADP 18 phosphate inorganic and 12 NADP will be released. Okay, so one more thing, this Calvin cycle is known as C3 cycle. Why it is known as C3 cycle? Because the first stable product of this cycle is 3-phosphoglycerate, which is 3 carbon compound, 1, 2 and 3. 3-phosphoglycerate is 3 carbon compound, hence the term C3 cycle is given. And this is the second phase of photosynthesis, which is termed dark reaction. So a dark reaction term is given, it doesn't mean it happens in the dark only. Just for the contrast, there is one thing, light reaction. So in the contrast, for the dark reaction name is given. Dark reaction depends upon the light, but not directly. The dark reaction, this Calvin cycle depends on the products of light reaction. So what are the products of light reaction? ATP and NADPH, which is used in this Calvin cycle or C3 cycle to fix the carbon dioxide molecule. So this was the Calvin cycle or C3 cycle or dark fixation of carbon dioxide or dark reaction or second phase of photosynthesis I have discussed in brief. Thank you.